Hello, in this video we will discuss about the urea cycle. So we will discuss about the biochemistry of urea cycle, also the overview of the urea cycle. What is that? So in mammal, urea production due to terrestrial environment which that is less water required for excretion of nitrogenous waste, because the terrestrial animal, for example the human being, which that is consume less water as compared to the fish and other water containing more water environment, for example the ocean, sea, as well as river. So in this way, the ammonia which that is required 500 ml water per gram ammonia requirement for the excretion. So it is impossible for the human. So that is why the conversion from the ammonia to into the urea is important. For the excretion, 50 ml water required for 1 gram of urea. So the excessive urea excreted from urinary system because toxic in our body. So in our body the detoxification will be occur from the liver. For the excretion into the urinary system is more important. So here is the liver, which that's contain two lobes, smaller and bigger lobe. So this liver contain hepatocyte we will discuss also later in this video. So here I am drawing the blood circulatory system. This blood circulatory system basically when during the eating time, here is the stomach, which that is when we will eat some food, for example containing amino acid or containing protein. So this food will, when we will intake, it convert into the bolus after the semi digestion of the carbohydrate, and that's go into the stomach and further digestion of the protein, and this digestion of the protein will convert into the chyme from the bolus and go into the duodenum, where the further digestion will be occur. That complete digestion will convert into the amino acid, and that amino acid will be absorbed into the circulatory system from the portal vein. So the portal vein will go into the liver. And after the excessive ammonia, so the excess ammonia toxic in body will convert after the metabolism. So the catabolism of the amino acid produce ammonia. This ammonia is more toxic, and here is the assimilation will be occur of the amino acid. For example, assimilation mean the part of the body of the food. So after this ammonia formation, after the catabolism of the amino acid, this ammonia is contain one nitrogen. So that's go into the liver. After the going into the liver, it will convert into the ammonia to the urea. So this here is I am drawing the one cell for understanding the urea cycle. So let's begin to understand the overview of the urea. So here is the 2 ATP and carbonic hydrogen bicarbonate and one ammonia will combine together in the carbomole phosphate synthase enzyme 1 used to produce the carbomole phosphate. This converts by the combination of ornithine to citrulline conversion by the fixation of carbamyl phosphate to the arginosuccinate synthase enzyme used to arginosuccinate lyase enzyme to produce arginine, and this arginine convert into the ornithine, and during the arginine to ornithine the water will be added to arginase enzyme to produce the urea. This urea will be excreted from the excretory system. Here is the kidney, ureter, bladder, and urethra. So that urine will be occur. But in the case of the dysfunction of the nephron will uremia formation. That uremia formation, the excessive urea in our body will give the toxic in our body to their metabolic disorder, and in this way our life-threatening problem can be occur. So let's begin to understand the biochemistry of urea cycle with detail. So first of all, I want to say this is the glutamate. First of all, we will understand that's glutamate is the amino acid which that is ionic, and this glutamate ion which that is the amino acid, and the glutamate dehydrogenase enzyme used to produce the ammonia, and this ammonia formation will be occur which contain one nitrogen. So after the ammonia formation, how it's possible? After the separation of the alpha ketoglutarate, this alpha ketoglutarate is very important for the Krebs cycle. Krebs cycle, so produce ATP, NADPH, and FADH2. And in this way, here is the ammonium ion. And in this way, here is you can see this is the Krebs cycle is important, and this alpha ketoglutarate is used for the Krebs cycle to produce energy for electron transport chain for different mechanisms. So this is the catabolism of the alpha ketoglutarate is used for the catabolism. So here is important thing is that after these two ATP used and hydrogen bicarbonate is used to separate the phosphate group and 2 ADP and proton. So the carbomole phosphate synthase enzyme 1, which that is used to produce the carbomole phosphate. And this carbomole phosphate is basically is the attachment of the phosphate group. Here you can see that with the carbomole. And in this way, important thing is that when go into the ornithine combination with the ornithine combination to the ornithine transcarbamylase enzyme used to release the phosphate group, inorganic phosphate group, and the citrulline formation. The citrulline formation is basically is the chemical formula you can see in this diagram to understand. So the aspartate is the another amino acid will bind with the carbon side, and this carbon side binding with the amine group, and in this way the structure will more complex, and in this way the citrulline in the presence of ATP to release AMP and pyrophosphate. In this way the too much energy used to arginosuccinate synthetase enzyme in the presence of arginosuccinate synthetase enzyme to arginosuccinate formation, and this arginosuccinate formation is look like this, and this chemical formula you can understand as well. So here is the when we will intake food, so the arginine is the basically is the non-essential amino acid also produced in our body, while the arginine is basically come from the food also. So it is also convert from the arginosuccinate into the arginine, and this arginine is basically is the arginase enzyme. This arginase enzyme is important for the conversion of the arginine into the ornithine, and during the conversion of the arginine into the ornithine, the water will be added to remove the urea. So this is the urea formation, which that is come from the ammonia. So the ammonia convert into the urea, and this urea is the 2 nitrogen containing compound, which that is absorbed less in water, so the requirement of the water will be less. But in the case of the bird, which that produce the uric acid, we will not understand. So the uric acid required more or less water for excretion. So the important thing is that arginosuccinate will be arginosuccinase enzyme used to fumarate production. So the fumarate is important also for Krebs cycle. So here is the ammonia, which that is during the liver cirrhosis will be occur. So the inadequate detoxification will be occur, and the ammonia will be more in our body, will damage the other part of the body, and the edema formation due to the excessive water, and the brain will be damaged due to the hepatic encephalopathy. So in this way, this is that there are many complications can be occur due to the urea cycle disruption due to the urinary system dysfunction. So thanks for watching.